You'll be my most.
Don't fart. That's what he's worried about. It's not the talking. Can I see your phone?
That looks like something. Because there's so much grass.
Here, there, wherever you want to be. Not way in the back. I think it'll see over. Oh, maybe it won't.
You better have written that in a secret place that you'll forget.
Okay.
I can do it. That way I don't have to worry about things. What time is it?
It starts at 1 right now.
Sheila and Peter Berger's maybe are having like Super Bowl. If I save this for family.
Not something that, you know, what happens in the middle, I'll come to you. Hang on to it. I got you.
Hi.
Steve, good to see you. This is my wife, Carolyn.
Hi.
Nice to meet you.
Nice to meet you.
You're a kindergarten teacher?
I'm a preschool teacher, actually.
Preschool, even
Yeah.
nicer.
Yes.
I thought I should meet you because one can never meet enough nice people. Are you nervous?
I'm nervous? No.
He's not nervous.
He's not, and that makes me not nervous. I'm just so proud.
I am too.
So, yeah.
Great. You're going to knock him dead, right, Dan? You're going to knock him dead?
I'm going to knock him dead. And that one back. But I can take it.
Thank you so much for your
Yeah, no, I can.
Is he going to take it?
It's definitely on the list.
I hope he takes it. Remind him that I want him to take it.
More intuition that there's somebody who understands and needs something. So he can get what, you know,
This is for me, not for him. Okay.
Well, I know he's very good at
I don't usually sit. I don't usually sit on this side of the room.
This is my computer.
All right, I think we're gonna get started. Hello. Yes, I'm Lockwood. So, it's really funny. I get a lot of questions from my students, like, why do you do this job? You have all this stuff that's kind of a pain, which is true, but there are some really great things about this job. Science is a lot of fun. But the truth is, getting a student to the point where they can do a defense and really, present a really finished project to you and to the rest of the community is really a sense of pride. It's a great thing to be able to do. It's one of the things that makes this job very rewarding. And Daniel Teasley is no exception. He has been a great member of the lab. And, those of you who know Daniel probably won't be surprised by this, but, when somebody sends you an email that says, what you work on is shit hot, I have to rotate, you kind of have to let them rotate. He sent me that email 6 years ago, 5.5 years ago, I didn't remember now, but, I still remember that specific email, and I was like, of course you're gonna rotate. And that attitude really hasn't wavered, and that is something that also makes this job so much fun. To see people come in, to be excited, to really take ownership of their own project. It's theirs. We're just there to kind of help smooth the edges out. It's really fulfilling, and Daniel has done that from the beginning. And of course, you have to always go to his friends to get the best picture so you can embarrass him a little bit. But, I think the thing that's great about these is it doesn't just show that they're progressing through grad school physically, as you all know, kind of things change, but more importantly, it's attitude. It's very, positive attitude, really engaging with the people around him and really enjoying what he does. And I think that, sometimes a little frightening even, but, some individuals, I'm gonna let him explain that at some point.
I'm the least embarrassing one.
Which is why you might stand out in this case. And then, of course, they often move on to even better things. And so, for those of you who don't know, I can't imagine who you are. Daniel was recently married to a wonderful woman sitting in the middle there. And I think that's just added a smile even bigger to his face. So it's really great to be able to introduce him to you. He's decided to have such a long thesis title that I had to bring it up so I wouldn't screw it up. And today what he's going to do is present a very interesting piece of work where he's really, taken a dogma. This, sometimes people get tired of hearing this word, but taking a dogma, something we thought we knew about protein, and really found out something new and very interesting that's making everybody within the lab, within the field, really rethink what they thought they already knew about basic programs such as DNA replication that was already supposed to be solved and in fact isn't. And so what he's going to present to you is his thesis entitled Flap-ended nuclease 1 promotes telomere replication and stability by distinct mechanisms on the leading and lagging strand. And with that, I will let him take over.
All right, we're good. Thank you, Sheila, for that introduction. It's very nice and less embarrassing than I expected. It's a good way to start out, I guess. Sheila, before we start and talk about the details of that work, I want to take a few steps back from this specific topic and try and frame the work in the right context so that you can hopefully appreciate why it's important. And to do that, I'm going to start with the topic of DNA replication. Now, all cells carry a genome, a genetic blueprint of information that encodes the instructions for that cell or an organism in the case of a multicellular organism. And when a cell wants to divide through this process of mitosis to produce two daughter cells, it first must replicate its DNA. And this has to occur because you want each daughter cell to have an identical and complete copy of the genome that the parent cell has. So you can't really think about DNA replication without thinking about DNA structure. And really, they're almost inseparable conceptually. One of the things that's so beautiful about DNA, and this is the original proposed structure from 1953 that Watson and Crick published, and it's pretty much accurate to this day. What's so amazing about this structure is that it's an ideal structure for copying. So if we take this double helix and we flatten it out and zoom in a little bit, I'm showing you here a piece of DNA that's replicated. What you can see is that there are backbones in blue, and these are made up of ribose sugars that have been connected together. And each sugar is connected to a unique base, adenine, thymine, guanine, or cytosine, A, T, G, or C. And these are the letters that make up the genetic code. Now, as you can see in the parent molecule that hasn't yet been unwound, A's are always paired to T's, and G's are always paired to C's. So in this way, we can use an enzyme, DNA polymerase, to synthesize a new copy off of one strand by base pairing the appropriate bases to the template. Now, one last thing that I want to mention is that the double helix is anti-parallel. And what I mean by that is that DNA strands have directionality. Here they're marked by the chemical notation 5' and 3', which doesn't really matter what that means. What matters is that it runs on the top from right to left, and the bottom from left to right. So the double helix is moving and twisting in opposite directions, essentially. Now, I showed you that DNA has directionality. What I didn't show you is that the enzyme DNA polymerase also has directionality, and it can only replicate DNA in the 5' and 3' direction. So what I'm showing here is a replication fork where a parent molecule is being opened up for replication. And because of this directionality that DNA polymerases exhibit, there are two separate processes that are going on here. On the top, I'm showing leading strand replication, and here, replication occurs in the 5' to 3' direction, and that's in the same direction as the opening replication fork. So this event, this replication, occurs more or less contiguously. It just keeps on going as the fork opens up. In contrast, at the lagging strand, the 5' to 3' synthesis direction moves away from the opening replication fork. And as a consequence of that, the DNA has to be synthesized in short, discontinuous fragments called Okazaki fragments. Here's the previously synthesized Okazaki fragment. This one is currently being made, and when the helix opens up a little bit more, there will be another one that lays down and gets made as well. And all of those pieces have to be put together in order for replication to complete the lagging strand. Now, I want to show you a video to give you a picture of what this might actually look like in the cell. Hopefully the computer is not going to upset me. It doesn't. So in here, I'm showing replication fork. Blue is the helicase unwinding the DNA. Here's the leading strand and the lagging strand. What you can appreciate is the leading strand down on the bottom is just moving this continuous process. The single strand's coming out, you're getting double strand down here. In contrast to the lagging strand, you're getting this kind of looping event where a piece is being ligated together, and then you start synthesis of another piece and it gets ligated together, and so on and so forth. So in large parts of your bodies where cells are dividing right now, places like the bone marrow or the intestinal lining, this process is happening in millions and millions of cells. But it's important to note that a video like this makes it seem like we know how this process happens from A to Z. Some have considered DNA replication as a problem that's been solved for a long time, but that's not all the truth. And that's not just something that people who work on DNA replication study. So what I'm going to show you today is going to outline the role of a particular DNA replication protein doing things that we did not expect it to do at all. And the take-home point is that replication is more complex than it seems, even based on an animation like this. That's not happening. So fortunately, we have a lot of new technologies and new research and continuing research that's enabling us to expand our understanding of this process. To get back to the process itself, replication, I want to really convey to you also the idea that DNA replication is extremely difficult to do. So in humans, the genome consists of 3.2 billion base pairs. That's 3.2 billion letters. And there's 2 copies of that genome in every cell. So if you take a single cell's genome and you stretch it out as a single linear piece, it reaches 9 meters high from the floor. So every time one of your cells wants to divide, polymerases in that cell have to copy 9 meters worth of DNA. And that's a lot to do. And it's especially a lot to do in light of the fact that human cells divide quickly. Often they can divide in less than a day. Sometimes they take much longer too, but we're looking at the minimum case here. Bacteria, which have much smaller genomes, so the amount that has to be replicated is a lot less. Replicate even faster, say less than an hour. So you think about having to copy a lot of material in an hour time scale. You've got to have a process that's pretty efficient. But the most fascinating thing is the accuracy at which this occurs. So the eukaryotic DNA polymerase error rate is about 1 error in every 10,000 bases that are synthesized. And that's pretty good. But if you then add on the DNA repair that occurs during and immediately following replication, that error rate drops to less than 1 error in 1 billion bases synthesized. So if you have a cell that divides, you might expect 3 or fewer errors that occur in that DNA. And that's remarkable to copy that much material with so little error. And this is what keeps things like mutations from accumulating too rapidly and causing diseases. Mutations do accumulate and lead to things like cancer. But the fact that replication is so high fidelity is a stopgap to prevent that in some ways. If it weren't hard enough to replicate DNA, there's some things that make it even harder in the genome. And I want to outline 3 of those things. One of them is repetitive sequences. And there's a couple different kinds of repetitive sequences. I've listed some here, retrotransposomes, microsatellites, and this TTH-GG is the vertebrate telomere repeat. That may be some foreshadowing for you. All of these things are repetitive. This last one in particular is extraordinarily repetitive. Like it makes the others look easy. So imagine reading a book with a 3-word sentence over and over and over and over again. And you'd envision that you might lose your place on the page. Much the same thing happens during replication. Repetitive sequences can do something called replication fork slippage, which can cause DNA damage. Now, secondary structures are also an important player, and these are structures that aren't the DNA double helix, basically. And some of them are formed because of repeats, like this G quadruplex structure is formed from G-rich sequences, so this can lead to that. And this is a T-loop, it's just another structure. But both of these things and other similar topological structures have to be resolved by enzymes, usually helicases, in order for the replication fork to move through and replicate. Now, the last thing that's a real challenge to replication is transcription. So transcription and replication, if they're not temporally separated, then they're occurring in the same template strand. And this can result in what we're showing here would be a head-on collision where the fork is moving to the right and the RNA polymerase moves to the left. The opposite's true here. This would be a co-directional collision. Now, I wouldn't have shown you all of these things if there weren't one place where they happen just to make things a lot of fun. And that place is the telomere. All of these things happen at the telomere and make it an extraordinarily challenging replication template. So what is a telomere? By the most basic definition, it's the end of a linear chromosome. And here what I'm showing you are sort of examples. This is a metaphase chromosome spread from human cells. And we've processed the chromosomes with a technique called FISH, which you'll see a lot throughout the talk. And what I've done is used a red fluorescent probe to mark the telomeres on these chromosomes. You can see, like this one, that has 4 red telomeres, and the schematic shows this nice and blown up. So telomeres are composed of a highly repetitive G-rich sequence, that TTA, GGG repeat I showed you on the previous page. And they can fold into things like G quadruplexes or T-loops, both of those structures I showed you. But that's what they are, not what they do. What they do is they allow the cell to make a distinction between a natural linear chromosome end and a DNA double-strand break. And the importance of this function cannot be overstated because a single DNA double-strand break in the cell is sufficient to completely arrest cell division. So it's really important that the cell is able to recognize between what's actual DNA damage and what's just the end of a chromosome. And there's obviously lots of ends in a single cell because there's multiple chromosomes. So in doing this role, they protect chromosomes from end-to-end fusions in particular, but also other types of instability. And they prevent genomic instability at large. So I want to give you an example of what happens when things go wrong. And what I'm showing here is an experiment in which the telomere protein TRF2 has been depleted of cells. And you can look, before that's happened on the left, there's some metaphase chromosomes with yellow telomere probes. And they look pretty much like the previous page. Everybody's happy. But as you deplete TRF2, you start to see some problems. And in particular, by day 6 and 7, you can see stretches of telomeres that are strung together, 4, 5, even 6 telomeres, 6 chromosomes strung together. And they fused at the telomeres. Now, if a cell were to try and divide with fused chromosomes like this, the chromosomes would break at the portions in the middle of the chromosome, say. And one daughter cell might end up with multiple copies of something, while the other daughter ended up with no copies of that same piece of information. So it's really important that this not happen. And this is a good example of how genomic instability is bad. So in order to prevent this, we have to maintain telomere integrity, and telomere replication is essential for DNA, for telomere integrity. So how do cells maintain telomere stability and replication? A large part of this comes from the network of proteins that interact with the telomere, for which I'm showing the schematic diagram here. Now, these proteins are all anchored by a 6-protein member complex called shelterin in blue here. And these 6 proteins are the keystone of all of the telomere, all the proteins that come to the telomere, so to speak. Now, you can see also there's a number of proteins that interact directly with the shelterin member, like telomerase, and then some that interact indirectly, like 1433 interacts with telomerase, interacts with TP1. So the network is big. Now, it was thought for a long time that the purpose of all of this, especially sheltering, but really the whole thing, was to hide the telomere, to disguise it from the cell, so that it wasn't seen as a double-strand break. So by putting a mask on it, so to speak, you could avoid that repair and maintain genome stability. But what's become increasingly clear over the last decade or so is that shelterin is doing something altogether different than that. This complex is actually recruiting, actively recruiting, in fact, these DNA repair and replication proteins to the telomere. And they do this to promote a sort of partnership in which the DNA repair and replication machinery are used to maintain telomeres when problems arise during replication. And it's important to remember that there's a lot of proteins on this list, and a lot of them probably do multiple things. And not all the proteins may get to the telomere at the same time. So even though there's redundancy in this type of network, there are still going to be cases where the telomere is going to be incorrectly replicated because all of the stars have to align for all of those mechanisms to work correctly. But today, I'm going to tell you about one particular protein that was identified by Sheila in a screen for telomere binding proteins. And that protein is flap-ended nuclease 1, or FEN1. What exactly does FEN1 do? It has several enzymatic activities. But the most important of these is a flap-specific endonuclease activity, which I'll show you here. You might remember this replication fork from a few slides ago. I want to zoom in on the lagging strand shown here at the bottom. Look at it over here on the right. Here we are replicating an Okazaki fragment, and as it nears the end of its track, it runs into the 5' end of the previous Okazaki fragment. Now, the polymerase doing this synthesis has an activity called strand displacement activity, which pushes the 5' end of that previous Okazaki fragment up into a flap. And this is where FEN1 does its work. It cleaves at the base of these flaps, which then leaves behind a nick that can be ligated together to complete DNA replication. Now, it's important to note that this is a process that should not be required of the leading strand because we don't have fragments forming discontinuously, so we shouldn't have flaps. Now, FEN1 doesn't just do replication, it also does repair, using the same flap cleavage mechanism in particular, and perhaps some additional enzymatic activities. It participates in base excision repair and a few other processes. But to get back on track, I told you that FEN1 was identified in the telomere interacting protein screen. And this wasn't surprising to us because the homologs of FEN1 in fission and binding yeast have known roles at the telomere in those organisms. So we wanted to understand what it was that FEN1 might do at the telomere in the mammalian cell. And so I'm going to start with some work done by a colleague of mine, Abhishek Saharia. And what Abhishek found was that when he depleted cells of FEN1 and examined the chromosomes for telomere defects, he uncovered a phenotype called sister telomere loss. And so in a normal chromosome, you have these nice 4 dots, all 4 telomeres are intact. What Abhishek saw was that in some cases we were missing a telomere from the end of the chromosome, which shown here. And this was a phenotype that was striking in part because it was so significantly increased. So he decided to look at this further. He wanted to understand better how FEN1 was preventing this from occurring. And the first thing that he wanted to do was to utilize a technique to try and identify if it was lagging strand specific or leading strand specific, given that FEN1 and replication has this big role in the lagging strand. Unfortunately, we were able to take advantage of a technique called chromosome orientation fish, or co-fish. And by processing chromosomes in a specific way, we're able to use strand-specific probes that are differentially labeled to identify telomeres that are replicated by either the lagging strand in red or the leading strand in green. Now, when this procedure works, normal chromosomes look like the 3 that I'm showing you here. You have 2 red telomeres that were replicated by the lagging strand, and 2 green telomeres that were replicated by the leading strand. So we decided to use this powerful technique to ask if FEN1 depletion induced sister telomere loss in a strand-specific fashion. And what we found was that that was in fact the case. So I'll walk you through this. If you look first at the chromosomes, you'll see that the ends here, where there's an arrowhead pointing, are missing a red telomere. This one we have green, red, and where's that other red? Well, it's not there. So if we quantify sister telomere loss events and quantify whether they're leading or lagging strand events, we get what's shown here on the right. We see that upon FEN1 depletion, there is no change in leading strand specific sister telomere loss. But upon FEN1 depletion, there is a substantial increase in lagging strand specific sister telomere loss. So next, we wanted to identify what FEN1 functions might be important in this particular role. And to get into that, I want to show you a structure of FEN1 and just very quickly point out a couple of features that are important. Up here where it says M1 and M2, these are metal binding sites, and this is the catalytic pocket of the enzyme. So this is where it's doing its dirty work of cutting DNA. But what I want you to focus on is way down here at the end of the protein. And there's 2 parts of this protein that are particularly interesting to us. One of them is a PCNA interacting peptide, or PIP box, marked as PCNA binding region here. And this portion of the protein allows FEN1 to interact with the replication sliding line PCNA. And it's essential for FEN1's ability to process Okazaki fragments. We then have this C-terminal tail of the protein, which has no structure. And the reason they didn't give us a structure is because it's an unstructured protein. So this part of the protein is known to interact with 20-some other proteins. And that's just what's been published. There's probably more. And the C-terminus has been implicated in Fen1's DNA repair activities. So when you use a C-terminal deletion mutant of the protein, it's deficient in DNA repair activities, but competent in DNA replication activity. So using the 7 mutants, we wanted to go and look at the sister telomere loss phenotype to try and understand if any of Fen1's functions were improved. As before, we see no changes at the leading strand, i.e. sister telomere loss, the specific leading strand is not changing in any conditions. Also as before, when we deplete Fen1 from the cells, we see a significant increase in lagging strand-specific sister telomere loss. Now, when we express the wild-type Fen1 cDNA, we're able to rescue that defect back to the control levels, indicating that this phenotype is specific to Fen1 loss. And unsurprisingly, when we use this DA mutant, which is short for D181A, this is a point mutant that is nuclease dead, so it's a null allele. And it's unable to rescue the defect, just so it looks the same as Fen1 deletion by itself. What was striking to us, though, is that when we use that delta C allele, which lacks the C-terminal region interacting with all those other proteins, you, again, fail to rescue this defect. And what this indicated to us is that it's Fen1's DNA repair activity that's important for preventing sister telomere loss at the lagging strand. Now, given that it's Fen1's DNA repair activity that's important for this, we wondered if Fen1 was important for DNA replication at large in human cells. So we carried out 2 experiments to try and directly address if Fen1 was involved in bulk DNA replication in human cells. And we did this in 2 ways. An S-phase progression assay I'm showing you on the left, we used HeLa cells that were controlled or depleted of Fen1. And we pulsed them and then chased them and watched as they moved through S-phase. And what you can appreciate is that their movement through S-phase is identical, whether or not they have Fen1. So at this level, Fen1 does not appear to be required for cells to move through S-phase. Now, we're showing on the right a large T antigen dependent in vitro replication assay. And what you can appreciate is that at each time point, when you start to see bands, the bands are of relatively equal intensity. So the scramble here looks the same as the Fen1 depleted. When you quantify that, you see here that the DNA replication kinetics in vitro between lysates from control cells or Fen1 depleted cells are identical. So these indicate that Fen1 depletion in human cells doesn't grossly affect DNA replication. And this is consistent with Fen1's role in budding yeast, where Rad27 cells can still divide and replicate their DNA. They have other defects and they would have trouble responding to stress, but at whole, they can do the job. So what we think actually happens here is that Fen1's replication-specific lagging strand role of processing for Kazaki fragments is compensated for by other nucleases such as DNA2 or EXO1, which possess similar activities. Now, in data I'm not showing here, we also examined Fen1 depleted cells for cytogenetic defects, and we found none. And this again provided additional evidence that Fen1 is largely dispensable for DNA replication in the human cell. Now, given that fact, and given the fact that Fen1's telomere stability depends upon its repair activity, we were curious as to what the molecular mechanism might be that would allow Fen1 to suppress sister telomere loss. And at the time, we were reminded of some recent work on the RECQ helicase work. And what Jan Karlsleger's lab had shown when examining this protein work was that if he expressed a helicase-dead dominant negative Warner allele in these cells, he saw all of a sudden a tremendous induction of lagging strand-specific sister telomere loss. So by removing one function from these cells, he's getting exactly the same phenotype that we did in our Fen1 depleted cells. And this was especially intriguing because Warren had also been recently shown to co-localize with Fen1 during DNA damage. And furthermore, the C-terminus of Fen1, which is required for the prevention of STL, as I showed you earlier, is the part of Fen1 that interacts with Werner. So this raised the possibility that Fen1 and Werner might be interacting together to do what they're doing. So importantly, Werner had been shown to facilitate this process of replication fork reinitiation. This occurs after replication forks have stalled and need to be restarted. And so given that Werner was important for that, and given all these other similarities between the two proteins, we wanted to ask the question, does FEN1 play a role in replication fork reinitiation? So we established an experimental paradigm where we treat cells with hydroxyurea for 16 hours to deplete them of FEN1. To deplete them, to arrest their replication forks. Instead of deplete FEN1 plot, sorry. So after 16 hours, all of the replication forks in these cells should stall. We then release the cells without any HU in the presence of BRDU. And if replication forks reinitiate during that 10-minute time period before fixation, they should appear as BRDU foci on BRDU immunofluorescence. And you can see in the control cells that is indeed the case. You see some nice green foci. They're not real bright because of the reactor, but they're there. And I think what you can also appreciate is they're not really there in the FEN1 depleted cells that we examined. It's to us that fork reinitiation is happening pretty well, but if cells don't have FEN1, they're deficient in that process. So we next decided to look at some more FEN1 mutants to tease out which of the functions of FEN1 was required to facilitate replication fork reinitiation. So we saw quantified here the control cells, when then depleted of FEN1, we see a decrease in fork reinitiation. We're rescuing that by expression of the wild type allele. And to get this out of the way, we're unable to rescue it with the DA nuclease dead allele. We're unable to rescue it with the other null allele delta P delta C. So that was expected. We are also unable to rescue it with the delta P allele. Excuse me. We're also unable to rescue it with the delta C allele. Which indicates to us that FEN1's DNA repair activity is mediated by its C-terminal protein-protein interactions. It is not required for this process. But what was striking to us was that the E160D allele looked the same. It looked the same as the delta C. So the last thing I want to point out is that delta P allele does rescue the phenotype, and this occurs because the delta P, which is replication incompetent, it can't participate in DNA replication. Fork reinitiation is it's distinctly more a repair event than a replication event. So even if you think of, well, this is replication fork reinitiation, the machinery between the two is different, and so that kind of makes sense why this allele is able to rescue the defect. But the delta C and the E160D cannot, so those activities are essential. So we then wanted to carry out the same mutant analysis, and doing it looking for sister telomere goals. So before I showed you that the delta C allele was unable to rescue sister telomere loss, thus the repair activity was essential for that process. When we go back and we look at the other mutants that we haven't examined before, we see essentially a mirroring of the reinitiation pattern. So when we deplete cells of FEN1, we get an induction of sister telomere loss, which we can rescue by the wild type. We can rescue it by the delta P replication deficient mutant. But we're unable to rescue it by the null allele, which makes sense, or the E160D allele. The E160D allele, I didn't go into detail about that. It's a gap endonuclease deficient allele of FEN1. And it's thought that this gap endonuclease activity is required for FEN1's ability to process structures that form during a fork reinitiation event. So for the purposes of this, it's kind of a more specific delta C allele. So given that I showed you before that the delta C allele was unable to rescue the defect, the E160D allele is unable to rescue the defect. This indicated to us that the C terminus and the gap endonuclease activity are required for FEN1's activity at preventing SDL. And furthermore, those are the same genetic requirements as those for replication fork reinitiation that I showed you on the previous slide. So given these data, we propose this model for FEN1's activity at the lagging strand. Most of the time, replication forks get to the telomere and stuff happens, but replication occurs normally and it's completed and everybody goes home happy. But stochastically, at the lagging strand, structures like G quadruplexes form. And these are impediments to replication and lead to replication fork stalling. When Fen1 is there, it facilitates replication fork restart to complete replication. And this is a process that depends upon its C-terminal protein interactions and gap and nuclease activity, i.e. its repair functions. But in the absence of Fen1, these replication forks that have stalled never reinitiate. And since there's not another replication fork coming from the opposite direction, because you're already at the end of the chromosome, you get a complete loss of that lagging strand replicated telomere, which exhibits as lagging strand sister telomere loss. Now, while we were carrying out our work on this phenotype, we kept noticing another telomere phenotype that we'd seen, but we never really quantified. And I say that because it didn't appear to be coincident with the sister telomere loss phenotype. But we became especially interested in this phenotype when we saw it first described in the literature. And the phenotype I'm talking about is telomere fragility. So a fragile telomere is what I'm showing you here. It's a morphological phenotype. These are normal chromosomes. Like you've seen on some of these previous slides, they have 4 telomere signals and they're nice round dots. A fragile telomere exhibits as a smeared signal or a doublet or something else abnormal. You can see lots of them here. And so it's basically anything that's not a nice round dot. And you might ask, why is this a fragile telomere? What's the terminology there? Well, I want to remind you that in multiple locations throughout the genome, there are well-characterized common fragile sites. And these sites are referred to as fragile because when cells are subjected to mild replication stress, such as treatment with low doses of imidacloprid, the cells exhibit preferential chromosome breaks at these particular loci. And so I'm showing you an example here in cells that have been treated with imidacloprid. This group has marked one of these common fragile sites with a green fish probe. And you can see in the left where the probe isn't, you can see there's a nick in that chromosome and a break completely here. So that's fragility. That's the expression of a common fragile site. It's not really understood why this happens. There's a few theories. But going back to the telomere, this phenotype is termed telomere fragility because it's induced in exactly the same way as a common fragile site, i.e. the finicolor treatment. And in fact, we can induce fragility by multiple mechanisms of replication stress. So if we delete TRF1 from cells, TRF1 is a sheltering complex member that is essential for fork movement through the telomere. We see telomere fragility. Similarly, if we look at ATR deficient cells, they also exhibit telomere fragility, presumably because they don't have intact replication stress response checkpoint. So our question then was, does Fen1 also induce telomere fragility? And it does. So when we quantify and control versus Fen1 depleted cells, we see an increase in telomere fragility events. Now, you might be able to guess where I'm going next, but as with the sister telomere loss phenotype, we wanted to further go after this by understanding if it was restricted to the lagging strand. We figured the SGL phenotype is a lagging strand phenotype. Fen1 is a lagging strand replication protein, and this is a replication stress phenotype. So we wanted to see if this was a phenotype that would exhibit on the lagging strand. So we carried out CoFISH, and we found exactly the opposite of that. So you can see there's some green fragile telomeres marked with white arrowheads here. This one's easier to see. And when we quantified the fragility and looked at the lagging strand, there was no difference in these two in the control of the Fen1 depleted cells. But we saw a significant increase in leading strand telomere fragility. There was no precedent or explanation for what Fen1 might be doing at the leading strand. As I told you in the introduction, at least in the case of replication, Fen1 is very distinctly a lagging strand replication protein. So we had to search for a phenomenon that occurs at the leading strand telomere to try and account for Fen1's role in telomere fragility. And here we came across an instance in which something that was thought to be figured out a long time ago turned out to be not true recently. So for decades, telomeres were thought to be transcriptionally inert. And it was shown in 2007 that despite all of those decades of thinking, telomeres are in fact transcribed into long non-coding RNA called TERA. And in subsequent work, like the work I'm showing here on the right, it's been shown that TERA are essential for telomere stability. So in these cells, when TERA are depleted, you see an increase in 53 BP1 foci localizing to the telomere. You see that as yellow foci down here. This indicates that without TERA present, you get an increase in telomere DNA damage. So this and other experiments have shown that TERA are important for the cell, and not just a spurious transcript. Interestingly for us, and this is not true in vision yeast, but it is true in mammalian cells, the TERA are only transcribed as a single strand. So if we look at a Norman plot for TERA back here, and we use a C-rich probe or a G-rich probe, we only see TERA hybridizing to the C-rich probe. And that indicates to us that all of the TERA species are G-rich in the mammalian cell. The conclusion we draw from that is if the TERA are all G-rich, they all must be transcribed from the C-rich leading strand telomere. And it's that leading strand specific transcription that became the target that we were going to go after. So you might recall I told you in the introduction that transcription can be a potent barrier to DNA replication. And the fact that transcription of the telomere only occurs using the leading strand template means that RNA polymerases and the replication fork moves in the same direction of the telomere. That's towards the end of the chromosome. Now, because the replication fork moves faster than a transcribing RNA polymerase, this sets up the potential for the scenario on the bottom here, where a replication fork catches up with a slower moving RNA polymerase, and they experience a co-directional collision event. Now, work in bacteria has shown that when this event occurs, when a replisome smacks into an RNA polymerase that's transcribing, the RNA polymerase leaves or is ejected. The mechanism by which that occurs is not at all clear, but it leaves nonetheless, and leaves behind the nascent, currently being transcribed RNA. The replisome then shifts forward and uses the 3' end of that nascent mRNA as a primer to restart DNA synthesis and continue along its way. So this was an interesting thing for us because of one particular part of this diagram, and that's the part I'm showing here. What's left behind by this collision event is a 5' flap that's connected to an RNA-DNA hybrid. And 5' flaps are something that Fen1 is very good at cleaving. So we hypothesized that these co-directional collision events take place at the telomere. And in the absence of Fen1, these flap structures cannot be resolved and accumulate. And that's what ultimately contributes to replication stress and telomere fragility. We decided to test this model by treating Fen1 depleted cells with the cyclic peptide alpha amanitin, which is an RNA polymerase II inhibitor that binds the polymerase and reduces its movement on the template, but doesn't eject it off of the template. So the idea was, if we use amanitin to slow down the RNA polymerase, we'll induce more collision events. And so we wanted to see if this would first manifest as replication stress and then as DNA damage potentially. And what we found was that both treatment with alpha amanitin and Fen1 depletion, shown in these two here, induce mild levels of replication stress, as indicated by phosphorylation of CHEK1 on serine 345. However, in Fen1 depleted cells that were treated with alpha amanitin, this replication stress response is substantially increased. And even more strikingly, in these cells, we see phosphorylation of the histone variant H2AX, that's serine 139. This indicates to us that there's DNA damage actually occurring in these cells. The DNA breaks are happening. So these observations supported the model that I proposed by showing the conditions which increase collision events, the alpha amanitin treatment, and putting the cells under conditions that decrease their ability to process the structures, that's removing Fen1, can induce replication stress and even a DNA damage response. And we next wanted to ask the question, does this manifest as telomere fragility? And so we quantified telomere fragility in cells treated in much the same way. And what we saw was essentially a mirroring of the CHEK1 step. So when we treat cells with, deplete cells with Fen1, we see an induction of telomere fragility as before. And when we treat cells with alpha amanitin, we see an induction of telomere fragility. You compare 1 to 3. If you do both, you get even more, the most in fact. And so this gave us the idea that if we're seeing the highest rates of telomere fragility in the worst case scenario, the model seems like it's starting to hold up to things. So we found evidence that if we slow down the RNA polymerase, we can increase replication stress and increase telomere fragility. We next wanted to address another part of the model, and that part of the model is down here. And the question we had was, is there evidence for this structure being in play in telomere fragility that's induced by these events? So we decided to search for that evidence by expressing the endoribonuclease RNA stage 1. RNA stage 1 cleaves the ribonucleotides, that'd be the red ones right here, in the RNA-DNA hybrid. And we surmised that if we were able to digest this, the flap would fall off more or less, and we'd achieve the same result as FEN1 could achieve. So we depleted cells in FEN1 and then expressed RNA stage 1 to see if it was sufficient to rescue the defect. We found, as before, telomere fragility induced by FEN1 depletion, comparing columns 1 and 3, is leading strand specific. There's no change in the lagging strand shown here to the right. Strikingly, when we take these FEN1 depleted cells and ectopically express ribonuclease stage 1, we return those telomere fragility levels to the control levels. And this indicated to us that the idea that an RNA-DNA hybrid flap structure is actually maybe the molecular origin of this telomere fragility, this type of telomere fragility at least. Now, we also wanted to come at this from a different angle, and that's from the beginning of the model, so to speak. So if RNA stage 1 ectopic expression is sufficient to rescue fragility induced by FEN1 depletion, we've also rescued fragility induced by increasing the collision events, i.e. treating the cells with alpha amanitin. So we found, as before, alpha amanitin treatment can induce telomere fragility. And strikingly, when we treat these, or when we overexpress RNA stage 1 in these cells, we again see a return to control levels of telomere fragility. And so together, these data show us that RNA-DNA hybrids contribute to leading strand-specific telomere fragility that's induced by FEN1. And that's because we're relying on the specificity of the RNA stage 1 enzyme, which, to our knowledge, only does things on RNA-DNA hybrids. So, having shown that slowing down the RNA polymerase increases telomere fragility, and that RNA-DNA hybrids contribute to telomere fragility, we next wanted to go back to the genetic experiments to try and identify which of FEN1's functions are important for this phenotype. Now, I'll remind you that it's FEN1's DNA repair activity as mediated by its C-terminus and its gap endonuclease activity that was required for preventing sister telomere loss on the leading strand. We anticipated that since telomere fragility is a replication stress dependent response, that it would be FEN1's replication activity as mediated by its PCNA interaction that would be important for this phenotype. So we carried out the analysis as before. There was no change in any conditions at the lagging strand in telomere fragility. At the leading strand, in these two separate experiments we're graphing together, we're able to show an increase in leading strand-specific telomere fragility upon FEN1 depletion. We rescue that defect by expressing the wild-type allele. And we rescue it by expressing the delta C allele and the E160 allele. That may not look like a rescue to you, but it really is a rescue from. So the delta C and E160D, those were required for FEN1 dependent suppression of sister telomere loss. They're not required here for the telomere fragility role. Most strikingly, however, we saw that the delta P allele completely rescues the telomere fragility defect, indicating that FEN1's interaction with PCNA is dispensable for its ability to suppress telomere fragility. So this is important for two reasons. One, it tells us that FEN1's roles in preventing sister telomere loss at the lagging strand and suppressing telomere fragility at the leading strand have completely different genetic requirements. So they're not only working on different strands, it's different activities that are being done. And furthermore, it tells us that we don't know much about the telomere fragility yet. So using all of these mutants to probe the functions that we know about for FEN1, all we've been able to show, in fact, is that we do need FEN1's enzymatic activity to rescue telomere fragility, because the nuclease del allele fails to rescue. So it has to be active, it has to be able to cut DNA. But it's not clear how it might be getting to where it needs to go. It's not getting there via the C-terminal protein-protein interactions, nor is it getting there via PCA. So with these data at hand, I want to propose the following model for Fen1 activity of the leading strand. Stochastically, collisions between the replisome and a transcribing RNA polymerase occur at the telomere. By some process that's not well understood, the RNA polymerase leaves, and replication is restarted, as happens in bacteria. Now, in the presence of Fen1, those 5' flaps are cleaved, which leaves behind a short gap and some ribonucleotides that can be repaired by the DNA repair machinery down the road. Similarly, if we overexpress RNase H1, it can compensate for Fen loss by chewing up this hybrid and achieving more or less the same goal. But in the absence of Fen1, there are not sufficient levels of RNase H1 for that activity. And what happens is these flat hybrid structures accumulate. Ultimately, they lead to replication stress, DNA damage, and telomere fragility on the leading strand. Now, there's still some work to be done here. We searched for a smoking gun for a long time. And the smoking gun for us was the RNA-DNA hybrids. We wanted to use an RNA-DNA hybrid-specific antibody to IP the sequence in these cells and see if hybrid levels go up in response to Fen1 depletion. We weren't able to make that experiment work, and we think the likely explanation is that the hybrid requires a very long stretch of RNA-DNA hybrid, and we think that the stretches occurring here are probably no more than 9 or 10 nucleotides. So from the technical standpoint, that's not an experiment we've been able to make work yet. We also don't understand why RNA-DNA hybrid flap accumulation or why replication stress in general leads to telomere fragility. And that's mostly due to the fact that telomere fragility is a morphological phenotype on a metaphase chromosome. So sometimes it's a smear, sometimes it's dots. What are those 2 dots if you were to stretch the DNA out and look? And until we know that primary structure, it's going to be hard to connect the molecular dots between something like this and how that ultimately leads to the phenotype you see on the microscope. I also want to revisit Fen1's role in the ligand strand to start to wrap things up, and emphasize that there's work that needs to be done here too. I hinted at the fact that we think Fen1 and urinary helicase might be working together to facilitate replication of the but those are not experiments that we carried out, and they're experiments that we could carry out using co-expression, Werner cells, etc. And similarly, we've proposed that the origin of the defect here may in large part be G quadruplexes that form on the lagging strand. And so one of the experiments that is attractive to do is to use a G quadruplex stabilizing drug that will make these structures more permanent, more stable, and unresolvable by a helicase, and see if using such a drug dramatically induces sister telomere loss. So coming back to this big diagram of all these protein interactions, I want you to keep in mind that we took one protein, flapping nuclease 1, which interacts with 2 shell-ring complex members. And we were able to discover 2 completely different mechanisms for how it maintains telomere stability. So how many of these other proteins might also have those kinds of scenarios where maybe something's been discovered about how they work, but something else is not yet figured out? And how many of them have been examined? Not really at all. And that's not even counting the proteins that aren't part of this network. There are obviously additional proteins that interact with the telomere that aren't on this diagram. The point being, this is a vibrant field. It's a field with a lot of future for things to be discovered. So to wrap up, I want to remind you, and I hope I've convinced you, of 2 roles for Fen1 at the telomere. And the first of these, Fen1 prevents sister telomere loss at the lagging strand by facilitating replication fork reinitiation. And it does this by using its DNA repair activities as mediated by its C-terminus and its gap and nuclease activity. In contrast, on the leading strand, Fen1 suppresses leading strand telomere fragility. And it does this by processing RNA-DNA hybrid flap structures that are produced following a codirectional collision between the replisome and the RNA polymerase. And with that, I have some official acknowledgments to get through. So first, I want to thank the funding sources that helped make this work possible. Sheila's R01 and the CMB training grant that I was on for a few years made this something that we could do. I especially, of course, have to thank the Stewart Lab, Sheila especially, and she'll get more thanks in a minute. But, Abhishek will also get more thanks in a minute. I want to point out Abhishek especially. Abhishek, for those of you who don't know him, which is probably most of the room now, he was Sheila's first graduate student, and he started the work on Fen 1 at Sister Telomere Loss. And I was fortunate that I was able to come to WashU and start in Sheila's lab and help him finish that work. And that was a great thing because then it was something I could springboard off of into studying telomere fragility. And the sum of our work allows us to show this beautiful dichotomy of Fen 1's functions. So without Abhishek, this would not have been such an awesome story that it is. I also want to thank Sankar and Mai. Sankar is another student in the lab, and Mai is an undergraduate who's now graduated, so he's no longer with us. But they both contributed substantial data to the fragility portions of the talk. I also want to thank my thesis committee members, Peter, Susanna, Barry, and Zhuangsheng. You've all provided different help at different times. It's all been welcomed and made a big difference. So from making the science move forward to making me move forward, it's been, I've appreciated every bit of it. I also have to thank our collaborators. Most of these are people here at WashU, and I won't list all of them because I probably missed a few actually. And I want to thank Bonnie Brewer at University of Washington, who helped us with a project that didn't work out, which you'll hear about in a second. So with that, I'm going to move into the more savvy acknowledgements. So first, I have to acknowledge that this is the most professional picture you've ever seen. It's getting better than that. Sheila took me in as a young graduate student, and about a month and a half after I officially joined the lab and signed the papers, she sent me to Bonnie Brewer's lab for about a month to try and learn this technique that we ended up not using. I did make the technique work in Seattle, and then we couldn't make it work in St. Louis. We were literally told that it was the water that was the problem. But Sheila's confidence to send me on something like that and to trust that I would be able to make something work if it was worked, at least, is a trust that she carried out throughout my graduate work. And she gave me a lot of leeway and a lot of freedom, but she also knew when to kick me in the butt to work on something. I actually didn't really want to study telomere fragility when I was trying to come up with a proposal. And she convinced me that I should probably finish the counts before I made that decision. I was wise to listen to her. Now, many of you know Sheila as an expert in things like telomere biology, lentiviruses, stromal support of tumorigenesis. I just want to share with you what I think is probably Sheila's most cited paper, and that's this one. The accidental entrapment of cats in front-loading Washington. She's the first author. Now, I've read this paper. I think the take-home to this are that you shouldn't put cats in washing machines. And maybe the front loaders are easier for cats to get in, but I think a cat can get into any washing machine. This is not actually Sheila Stewart, this is Sarah Stewart. Thank you, Sheila, for taking me from the beginning to here. Made a huge difference in my life. I also have to thank Avi. I mentioned his scientific contributions. Some of you might not be surprised at all to find that the only picture I can find on Facebook with both of us tagged in it is of us drinking. I believe Adam has spilled beer on somebody's feet here. I'm not sure what happened, but that was pretty typical at that time in our lives, I'd say. This is probably a better picture of Abhishek to remember about. He was a mentor when I came into the lab, and he was an outstanding mentor. And, his mentorship is something I based my mentorship on, and getting me started in that way was critical for my career here. I also, of course, have to thank the lab. This is a pretty recent picture of us. Some of us have slightly left, like Elise and Mom, even though they're still kind of around. But the lab made it a fun place to come to work every day. They were supportive scientifically, they were supportive in other ways too. So they aren't just colleagues, but they're friends because they all came to my wedding, or they just wanted free food. I'm not sure which. I hope it was the wedding. I also have to acknowledge people in the lab who aren't with us anymore, two in particular. Ben Dow over here in the corner, and Julian Dawson here in front. they also made substantial contributions to my time here at WashU. I couldn't have picked a better group of people to work with. And so I'm just very happy to have been able to spend my time with all of you. I also have to thank 3 of my trainees. These are my 3 undergraduate students. This is Jane on the left, who was my first WashU undergrad. And she was a full-timer, so to speak. She went through the semesters on the summer. Adrienne was a summer student, and who's here, I believe, was my most recent undergraduate student. And having these 3 students made me grow in many ways as a teacher and as a scientist. And I would argue that I learned more from them than they learned from me. And I'm just so happy to see each of them succeeding so much in what they want to do. There's an MD PhD and a PhD and a future MD PhD trainee right there. So I'm very proud of all of them, but I'm thankful for what they brought for me to the experience. I have a lot of friends to thank. I won't go through everybody, but you might know there's a wedding picture theme in here. I guess that's what a lot of people who are important to you show up. This is everybody I went to college with, I think. And they all came to the wedding, and it was wonderful. I have to thank especially this person, Madeline and Christine, for being strong emotional support through the early years of graduate school. My friends Andrew and Betsy, and Justin and Liz, who are, I'd say they're Carolyn and my grown-up friends now. But whatever that's like. I also met a lot of people at WashU, of course, and we had a lot of fun at different times. As you can see again here, earlier pictures tend to include drinking. This is an entertaining story, but I don't know what it was. This one I want to stop at for just a moment, because Elise is here somewhere, I think. So you can do after the presentation. You can do it before and after. And it's like a 2-term president, before and after. Thank you all for being my friends and being supportive while I was here. And I especially have to thank these 2 people, Brandon and Jesse, who have moved on to San Diego. And they've been my closest friends here. Will continue to be my close friends for many years to come. Those of you who know my wife might know that she's one of 6 children, all shown here. And so I have been fortunate to inherit quite a few brothers-in-law and sisters-in-law. And we've had a lot of fun doing a lot of different things, whether they're not all baseball games, but holidays together, celebrations together. And I especially have to thank my father-in-law, Bill, and my mother-in-law, Anne. The last picture might not have loaded. There was another picture. There it is. So this family made me feel like I have a family here. I obviously have a great family. I'll talk about it in a minute. We weren't here, but they've made it feel like home. They've made it a place where I want to stay. My family, of course, gets a lot of credit for having the common sense to send me way far away so they didn't have to deal with me. Whether it's my dad's side of the family and my late grandparents, not pictured here, or my mom's side of the family and my late grandfather, not pictured here, to my older brother. And those of you who have an older brother will know that I don't need to say much about that. There are early negatives and infinite later positives. And my sister, who's against my advice, decided to pursue a career in research. But we can co-resurrect together. And of course, my dad, who is the first person who taught me that science could be interesting. A long time ago, he was working with orthopedic implant manufacturers, and an artificial knee will get like a 6th grader pretty excited about science, that that's something that can work. So that's where things started to take off. And I thank my mom for making me the smartest person I know in the ways that matter the most, which is to say, not any of this, but the ways that matter the most. I love you both, and I'm so glad you did. And I save the best for last, and that's my wife. We also don't only go to baseball games. Sometimes we hang out with a dog that's not actually a dog. And we have holidays together, and Then we had the best event this past year. The best event in the past 12 months will not be this defense, which is a good thing because I got married 7 months ago. So. And there's nothing that will surpass that, no matter how great publishing a paper or getting a PhD might be. And the more important fact, I think, is that publishing that paper and finishing the PhD might have been impossible without her standing by me, without her encouraging me and supporting me. So with that, I will take any questions that you might have.
So before the committee gets to drill him one last time, I just want to let everybody know that after the drilling is completed, there will be dessert, beverages, drinks over on the 7th floor of the BJICH. But before that, do you have any questions for Danny?
Dan, do you think it's just that it's easier to detect these things in the telomere tools, or do you think that this sort of damage is not happening elsewhere in the chromosome? It seems like those sort of mechanisms can also happen.
We think it's happening everywhere. So anywhere a co-directional collision can happen, it should leave behind that same structure. I think for us, the reason the telomere was attractive is because it provides an easily quantifiable metric, like a downstream look at here it is type of flag signal. But I don't think that that particular activity, the fragility suppression, is restricted to the telomere.
Can I ask a stupid question?
There's no stupid question.
When you have this replication, transcription collision that's resolved by FN1, by clipping off the cell by a prime fragment, you end up with this, what you depicted as double-stranded DNA with a little bit of RNA. Is that functional? Is that bad?
So it has to be removed, either by RNases H1 or 2. And so ribonucleotides in the genome are bad, and they are usually pretty efficiently removed. So it can't stick around like that. But presumably, it's better than flat. Hard to say.
Because it's not fragile.
Yeah.
You tried to pull down the IP with the DNA RNA antibody.
Yes.
Have you tried it with like an RNase that's catalytically inactive?
So that's our next step, and you can talk to Sankar about that. There's an even better approach. I think somebody's made a construct of the hybrid binding domain of RNase H1 alone that's conjugated to GFP. So you could then just do the GFP IP and take it out. And RNase H1 requires something like 9 bases, 10 bases to see a hybrid. So that would be a more feasible approach. And if I was going to be here for a long time, I'd definitely do it. Maybe somebody else. See.
If there are no other questions, we hope to see all you in 40, 45-ish minutes.
Due cafeterie, café,
Stop it.